Hello, and welcome to Virtual Investor Conferences. On behalf of the Life Sciences Investor Forum and our co-host, Zach Small Cap Research, we're very pleased you joined us for our quarterly conference. Our next live presentation is from FibroBiologics. And FibroBiologics is a cell therapy regenerative medicine company developing a pipeline of treatments and potential cures for chronic diseases. Please note you may submit questions for the presenter in the box to the left of the slides. You also may view a company's availability for one-on-one meetings by clicking Book Meeting in the top toolbar. At this point, I'm very pleased to welcome Peter O'Hearn. He's the founder and chief executive officer, and David Bouts, a senior analyst with Zacks Small Cap Research. FibroBiologics trades on Nasdaq under the symbol FBLG. Welcome, Pete and David.
Thank you for having me. Appreciate it.
Thanks. And thanks, Pete, for being here today. So I guess we jump right in. This will start general overview. So what exactly are fibroblasts? Why develop them as therapeutics? And what qualities do those cells have that kind of lend themselves to being suitable for therapeutic use?
So there's two cells in the human body you can use to regenerate tissue and cure chronic disease. There's a stem cell, which everybody's heard about, and there's a fibroblast, which most people haven't heard about. The irony is that fibroblasts are the most common cell in the human body. They actually outnumber stem cells 5,000 to 1. We like them better than stem cells. They're a more stable platform than stem cells. They're easier to harvest than stem cells. They have higher immune modulation, higher therapeutic capabilities. Stem cells want to be anything other than a stem cell. They're an inherently unstable cell source. They're a fantastic cell source, as we've seen. A number of companies have formed around those and are developing therapies. But compared to fibroblasts, they are a much more unstable cell source, much more difficult to work with. Fibroblasts are already fully differentiated into their what they're going to be. And stem cells want to be anything other than a stem cell. So fibroblasts can turn into almost any other type of cell, but you have to talk them into it. So it's a more stable platform. And being the most common cell in the human body, they're always going to be a less costly alternative to stem cells.
Okay. So why do you believe that fibroblasts are the key to treating chronic diseases?
Well, we've done the research. We've looked in the last 60 years of big pharma, no chronic disease has ever been cured. I mean, think about the trillions of dollars that have been spent trying to find cures for chronic diseases. It's because we believe that a man-made external chemical compound will probably never cure a chronic disease. In fact, if it was going to cure one, we think it would have done it by now. So a biologic defect in the body is where you get your chronic disease from. And we believe it's going to take a positive biologic process created through fibroblast or through a stem cell, through immunotherapy, or through gene therapy. So the three ways we think that you'll be able to cure chronic diseases going forward are cell therapy, where we are, gene therapy, which is CRISPR, and immunotherapy, which companies like Bristol Myers and those guys are involved with. Oddly enough, the last three large Nobel Prize winning technologies were originally in 2012. Stem cells, one for induced pluripotent stem cell, that's taking one cell source and turning it into another. Second hour of the story, he used a fibroblast to do that. So fibroblasts actually created the stem cell revolution. After that, you saw Dr. Jim Allison at MD Anderson in Houston develop immunotherapy for cancer, and he won the Nobel Prize for that. And then more recently, Jennifer Doudna and Emmanuel Charpentier won for CRISPR. So you saw that, and they just launched, just got approved for their sickle cell anemia therapy last December. So I think that you'll see the cures for chronic diseases come through those three platforms.
Okay. Where, for the company's purposes, where are the fibroblasts that you use derived from?
So literally they're skin cells. They're human dermal fibroblasts. We can pull them right out of your skin. So it takes a small dermal punch. So if you went to your dermatologist and had a mole taken out, it would be about the size of that, about the size of a number 2 pencil eraser. And from that, we can deliver over 200,000 doses of fibroblasts. Literally, from about the size of a pencil eraser, you can almost regrow the whole human body with fibroblasts.
So are these cells derived from each patient, or is this more of an off-the-shelf therapy?
So it's an off-the-shelf therapy. I think that when cell therapy first started, deriving it from the patient, which is considered autologous, was kind of the pathway that everybody took, and they were heading down that pathway. But when you think about it, there's only one location where they make Tylenol and only one location where they make Advil. They do that because they want to be able to assure the safety and quality of each run. You don't charge 200 facilities across the country with making those particular pills. Well, same thing with cell therapy. If everything you did was from that patient, you would end up with 200 facilities trying to come up with the same type of cell therapy. And you can imagine the clinical conditions are different, the people are different, the equipment is different, ambient conditions are different. So I think what you're going to see is allogenic, which is a off-the-shelf donor. So from a donor to another individual. What we see with fibroblasts by using that allogenic is that my fibroblasts work in your body the same as your fibroblasts work in your own body. And that's unusual, and that's unique to fibroblasts. They are a bit of hidden from the immune system. So we can have a very easy off-the-shelf solution using fibroblasts.
Okay. Can you talk about some of the challenges, obviously aside from getting positive clinical trial results, that need to be overcome to make fibroblast therapy become a reality?
So the challenges we are dealing with is a first-time technology. I mean, what I tell our scientists, everything that they do is the first time anyone has ever done it. So when we go to Mars and take that first footstep on Mars, it'll be the first time any human's ever done that. We do that every single day in the lab because nobody has pursued fibroblasts therapeutically. So everything we do is the first time. So creating science from scratch is really interesting. It's really challenging. Educating the market, the capital markets about what we're doing compared to stem cells and having that access to capital is challenging, and just the normal challenges with being a small public company.
Yeah. Okay. And I know you've talked before about the company's IP, and maybe you can talk a little bit about why having such a robust IP portfolio is so important.
Yeah, so we've spent a lot of time, commitment, and treasure on our intellectual property. We have more intellectual property with fibroblasts than the rest of the world combined. So it's a really important component of what we're doing. And while we were early on patenting methods to treat certain diseases, now we are adding to that our manufacturing process so that we can create a more efficient, more effective, more consistent product. For example, we started off with using single cells, and now we use spheroids, which are a collection of fibroblasts into one droplet, if you will. So there could be up to 3,000 fibroblasts. We like that because that it tends to have more staying power in the therapeutic area, so we have more time on target. In our wound care program, that spheroid literally has a very predictable biologic time release, and it kind of dissolves into the wound, so it has much more time on target. So as we are developing working on our existing IP, we're also developing new IP around the manufacturing process, different delivery systems, and kind of looking over the horizon at where we're going with those.
Okay, sounds good. So I'm going to dig into a little bit specifically about your products you're developing. And I know we're starting with diabetic foot ulcers. So I guess the real question is, why start there?
It's a great question. Fibroblasts are used in involved in every aspect of wound healing. So if you looked at a wound, you would see a lot of fibroblasts. What we can do is take take those fibroblasts, we can concentrate them, and we can have them on that target a lot longer. We think we can have a really excellent result with diabetic foot ulcers. We tested ourselves against the #1 product in the market, Graphics by Smith & Nephew, and we saw almost a 50% reduction in the amount of time it took to close that wound compared to the #1 on the market. So that's very exciting for us. What we want to do is to show the capability of fibroblasts early with something like those diabetic foot ulcers, and so we can get the market aware of the capabilities of fibroblasts. So we started with something that had, we believe is going to be successful. It has a short endpoint of 12 weeks instead of some of the other chronic diseases, which might have an endpoint at 12 months. So I think we're going to be able to have some really good results in short order.
Okay. And speaking of the results from that study, so what outcome is going to be most important in there? Is it how quickly the wound heals? Is it what percentage of each wound? What are you going to be looking at specifically?
Actually what percentage of the wound closes over what period of time, and also the quality of that wound closure, because we don't want it to reappear. So those are the big elements. You want to see those sides coming together on that wound. You want it to close quicker than what you would get with another therapy, but you also don't want that patient to readmit with another wound at the same spot. So it's the quality of the closure as well. Okay.
And you mentioned the preclinical results that you looked at with graphics. So for the clinical trials, are you going to have a comparator in that trial as well?
We don't. We're going to compare against existing therapies out there who've already done their clinical trials. So we're not going to do that.
Okay. So assuming you get positive results from this initial study, I guess what are next steps for the program, and what would be a time frame for those steps?
So we are planning to issue results on that this year, this calendar year. Then we'll move into phase 3 commercialization right after that for the diabetic foot ulcer. So early next year, you'll see psoriasis coming right behind that.
Okay. And okay, so you said phase 3. So you're going to finish the study this year, and then you can go straight into phase 3?
When we show our data to the FDA, that's what we'll be requesting. Yes.
Okay. And you just mentioned psoriasis, which is a good segue, because I was going to ask you about why psoriasis. Why do you believe it's amenable to being treated with fibroblast-based therapy?
Well, psoriasis, again, it looks like a skin condition, and it certainly is that, but it's actually an immune system condition. And so we have a tremendous efficacy against the immune system with fibroblasts. We saw it, which kind of led us into psoriasis with our multiple sclerosis trial. We did a small human trial for safety with 5 patients. What we saw in all 5 patients, well, it was only 5 patients because we're the first people that have ever injected fibroblasts intravenously into another human, was that we stopped the progression of the disease in all 5 patients. So we really think we have something in multiple sclerosis, and psoriasis is an immune condition. So we have seen in the lab, in our animal trials, we've done extensive animal trials of psoriasis, and it's really remarkable the results we're getting off that. In multiple sclerosis, we've actually seen remyelination. I don't know if anyone's claiming that. That's repairing the damage to that myelin sheath. With multiple sclerosis, you get a fatigue in that myelin sheath, you get breakage in that, and we've actually shown that we can repair that myelin sheath. So very exciting.
Okay. And so actually this question is for both the psoriasis and the multiple sclerosis indication, is that there are successful treatments out there already for both indications, but what advantages do you think a fibroblast-based therapy has over those currently available therapies for each indication?
So when we look at the existing therapies that are out there, they either overstimulate the immune system or they deplete the immune system. Those are really the two different ends of the spectrum that those treatments work on. And what you see in psoriasis and multiple sclerosis is a depletion of the immune system, a suppressing of the immune system so that the body can heal itself. What fibroblasts do is they do it without suppressing the immune system. So you don't have any of the complications that you get or any of the risk factors that you see when you suppress the immune system. Of course, you get a long list of side effects and risk factors that come along with that. With fibroblasts, we bring you right back to homeostasis, right back to the middle, and allow the body to heal itself without overstimulating the immune system or without suppressing the immune system.
Okay.
During COVID, I heard a lot of people say, wow, we just need to engage the immune system for COVID. I heard a lot of people on TV saying that, and I used to think, be careful what you ask for. An over-engaged immune system starts to attack things around it that you may not want.
Yeah, no, that's very true. So where do you think the fibroblast-based therapy would fit in the treatment paradigm with what's currently available?
Well, I think you would see it effective against the very similar treatments that you see with stem cells. Only I think it will be more effective, a quicker result, and I think it will be a lot less expensive than what you're seeing with some of the stem cell therapies. But you'll see it in all chronic diseases. I think we'll see good results in everything that we're working on.
Okay. Will head-to-head trials, do you foresee those being necessary for either indication with what's currently available?
We do. We did head-to-head trial for diabetic foot ulcers with the #1 player in the market, Graphics. We do compare to stem cells in everything that we do in the lab, so we always run a comparator using stem cells, and we have outperformed stem cells on every test we've done.
Okay. So kind of related to psoriasis, do you see any applicability of fibroblasts to treating eczema?
We haven't worked in that area. We just have to look at it and do some testing in that area, but we haven't worked there. But I think you would see results. I think the top one out there is Dupixent that's used for eczema, and that has a depletion function to that. So I think you would see some benefit from it.
Yeah. Okay. Coming back to multiple sclerosis for a second, you did mention a little bit about the phase 1 trial. Maybe if you could just talk a little bit about more about those results, and then what the follow-up is to what you saw in that study.
I'm sorry, ask that again.
So for the multiple sclerosis indication, you did previously mention that you had results from a small phase 1 trial. I just talk a little bit more about those results, and then I guess more importantly, what's the next step for that program?
Okay. Yeah. You know what was exciting was that we saw no further progression of the disease during that trial. We felt like we had seen remyelination, but it's very difficult to see that in a human. But we went back into the lab and we've done extensive animal work, and we have seen full remyelination with those. Now, we can't tell you the exact mode of action. We believe it could be some sort of a paracrine effect, or it could be that we're able to take the immune system back to homeostasis and allow the other healing areas of the brain to take over. So we're still working on the mode of action, but we saw it in all 5 patients, and we had no adverse events reported for that study. So it's really exciting. The fact that we were able to take allogenic fibroblasts, inject them into another human intravenously, and get those kind of results was really a game changer for us.
Yeah, absolutely. So turning to some of the earlier stage programs, one of the ones that you've mentioned before is kind of an extension of life through thymus regeneration. So I guess, can you discuss exactly what that is? Like, what's the theory behind it? And then how close is the company to potentially getting something into the clinic?
So it is a very exciting program. I like to sometimes say that it's exotic. The thymus gland is your teaching center for the immune system. You've heard of T cells, those are thymic cells. And as you get older, the thymus gland starts to shut down in a process called involution. By the time you're in your 60s and 70s, you almost have a non-functioning thymus gland. So you don't have more cancer in your 70s than you do in your 40s. It's just that your thymus gland stops teaching the immune system. So our original theory was if we could stimulate the thymus gland to regenerate its capabilities, we could allow humans to live another 15 or 20 years. What we've done in that process is develop an organoid. An organoid is a collection of cells that function very similar to the larger organ itself. And so we've been able to develop an organoid of thymic cells. We have injected it sub-Q. It's remained durably fixated for over 62 days, and it developed T cells during that process. So it's really exciting. If we could determine the thymic function and then base our dosage of organoids on that, we could theoretically inject sub-Q organoids to make up for that thymic loss, and you could keep people alive in a quality of life too, because it would have a systemic impact. Continue to teach the immune system.
Okay, and recently you put out a press release about using fibroblasts to potentially modulate mitochondrial activity against this probably earlier stage stuff, but I'm just curious if you could talk a little about how that would work and what's the mechanism behind that.
That's very, very early stage. So we're just beginning our proof of concept in that area. We'll have more to say on that probably in the fourth quarter of this year, but it's very exciting to use the thought that we could use fibroblasts to increase mitochondrial performance in the cell. It has a lot of implications across multiple platforms.
Okay. So looking at the pipeline as a whole, what program do you see potentially making it across the finish line first, assuming positive results, of course?
I think you're going to see wound care. I think you'll see diabetic foot ulcers. We'll see approval of that first. I think you'll see psoriasis probably right after that. We're expanding our wound care and beginning work in burn victims. You'll see surgical post-wound treatment for that. And then eventually there is possible to have a retail component associated with our wound care. So that is a platform inside of a platform.
Okay. So a little bit company specific. So what is the company's current cash position? And then how long will that cash run for?
Well, we reported $8 million at the last quarter, and we're working on our 10K now. So we'll be reporting that in I believe it's next month. In year end, though, we added an additional $10 million through a placement facility with Deep Oral in Yorkville. So we added that cash position, but we haven't reported our year-end cash position for another month. Okay.
And can you talk a little about the financial surrounding fibroblast therapies? I guess I'm looking more at cost of goods, that type of thing.
Well, we like to say I can't imagine any cell therapy being less expensive than fibroblasts. You're taking a small dermal punch. Stem cells are difficult to harvest, especially if you're using autologous stem cells. You either get them through liposuction or you get them through a bone marrow aspirate. Very difficult to harvest, difficult to keep alive, difficult to work with, difficult to expand. Fibroblasts, you can literally use a dermal punch. You can pull out a core from the skin and you can pull out all the fibroblasts you need. So it will always be a much less expensive harvesting technique.
Okay. So for you personally, I guess, how did you become interested in fibroblast therapy? Like, what was your aha moment where you said, okay, this is going to be really big and something that I want to be a part of?
So we originally had worked with a French neurosurgeon, a French dermatologist who came up with the idea of using a fibroblast to use in degenerative disc disease. So you inject it into the disc, and the intermittent hydrostatic pressure applied on that fibroblast by the vertebrae above and below it would force that fibroblast to turn into a chondrocyte and regrow the disc. That was the original provisional patent that they had filed. We acquired that from them. We were fascinated because we've worked on other technologies, but they were improved technologies. And this one was, if it worked, was a game changer. And so we put that with Rice University, and they developed this tabletop unit that would mimic the force of the spine, and it worked. Worked the first time, and it worked every time. So then we went to Rush University in Chicago when we did extensive animal studies using that principle, and we had success in those animal studies. While that was going on, we began looking at other areas, and I started challenging our scientists. I said, this looks like it might have a broader impact across other diseases. And it became an explosion of discovery. Literally, we were filing 3-4 patents a week. Every area we looked at we were seeing an impact using fibroblasts. So we began to develop a theory that the reason we've evolved with fibroblasts being the most common cell in the human body is probably because it's involved in every biologic process in the human body. And so while everybody was chasing stem cells, we were chasing fibroblasts, and we couldn't find anything that it didn't have a positive impact on.
Okay. So I guess in the last couple of minutes that we have here, let's go over why you believe that FibroBiologics is an interesting investment opportunity right now.
Well, I think that we're the only company in the world using fibroblasts therapeutically. This is a space that we have the most intellectual property. We have more patents in this space than the rest of the world combined for fibroblasts. So we have a first mover advantage. We have high protection around the products that we're developing. We have a very energetic, creative scientific team, and we have a relatively small market cap at this point for people to get involved, and be a part of our growth.
Okay, and we did have a question about the milestones. So what should investors look for, say, for the rest of 2025? What are the milestones?
So you're going to see our diabetic foot ulcer trial begin. We're doing that in Australia. That will begin in a May-June timeframe. You'll see interim results from that. You'll see early safety results from that, and you'll see our endpoint report from that this year. You'll also see at least 1, maybe 2 IND filings this year. That's exciting, and you'll see an expansion of our human longevity program this year. So we're very busy.
All right, sounds good. So Pete, thanks for joining us today. This was great.
Thank you for having me. I appreciate it.